We want to move now to one of our other scientists and speakers for some more breaking research that is happening. And I should mention, as I go on to Professor Jozef Gecz, that one of the hallmarks of our experts is that they have long, chunky titles. And these are sometimes hard to get through. So if I stumble, I hope you're going to forgive me. So here we go for Professor Jozef Gecz. Professor Gecz is National Health and Medical Research Council Senior Principal Research Fellow and Channel 7 Inaugural Chair for the Prevention of Childhood Disability at the University of Adelaide and South Australian Health and Medical Research Institute. Take a breath here. It's a big title, but it's a real title and it involves a lot of things. And Professor Gecz, who I'm now going to call Jozef for the rest of this conversation, has been working for 30 years in competitive research in genomics and molecular biology of childhood onset neurological disorders across Europe and across Australia. He has discovered and contributed to the discovery of more than 200 different genes. Quite a track record. He has also pioneered research into the genetics of non-syndromic intellectual disabilities and X-chromosome linked forms, and has expanded now into childhood genetic epilepsies, which, Jozef, is why I'm delighted that you are here today to talk to us about your latest research. And I also want to just mention, because I like chunky biographies, they tell us how smart you are and how much you've achieved. His achievements have been recognized by the Australian Academy of Science and the Australian Academy of Health and Science. I think I've got all that right, and many, many more. So apologies if I've left anything off, Jozef, but may I just say we're delighted that you're here with us today, and we very much look forward to your presentation.
Thank you very much, Sarah. I trust you can all hear me well and you can see my first slide. Now, it's really my privilege to participate to this conference and to talk to you. I've followed this conference now for at least a couple of years, and I was really amazed by the amount of information and the collective learning we all do. We learn from you, from the parents and advocates, and we learn from fellow scientists. So I hope today I will tell you an interesting story which is more focused on one specific gene, but I trust I will have other messages which many of you can take home after my presentation. So I'm going to talk to you about a very specific type of epilepsy as it was initially described, PCDH19 clustering epilepsy. It's a specific genetic epilepsy, probably one of the top 5 most frequent genetic types of epilepsies. And I've been privileged to work with 2 patient group advocates, and that is one led in Italy, established in 2012, with Francesca Squillante and her team on the left, and on the right, Julie Waters and her team at the PCDH19 Alliance in the United States. So I sit in Adelaide as a very nice medical school, and was in really a $5 billion medical precinct where we just try to change the lives for better for people living with different disabilities. We have been very fortunate during COVID time because we literally lost maybe 1 day of laboratory work over the last 16 months. We are well connected internationally, and that's really the basic approach of our research, to involve patients, other scientists internationally, because without that approach, really we would be on a back foot, just to say the least. So why we are different, and you keep hearing about it, maybe different reasons in my own experience, can be nutrition, can be which part of the hemisphere we live on. Like in Europe, the northerners tend to be taller than the southerners. Can be hard work or hard exercise, but we also know that the explanations are often in DNA, or as I will mention also here, something which sit on top of DNA, and Steve alluded to it in epigenetics, or for short,
Jozef,
epidNA.
Jozef, I'm so sorry. It's Sarah interrupting here from Melbourne, and I hate to interrupt you, but I wanted to let you know, and maybe it's just me, but I do not think we're able to see your slides. This is the challenge. We're all participating virtually. So let me just see if I'm being told that I'm not the only one. Let me give you a moment to see if you can get the slides.
My fault. Now you can,
Very good. I think there we go. Thank you so much. And I am not going to interrupt you again. I just know that everybody's going to want to see them. So I will be silent and wait until the end.
Please do. It's my fault. I got too excited. I forgot to press the share button.
That's all right. You go right ahead. Carry on. We've heard everything. Everything has been heard. We just didn't get to see. So we'll let you carry on.
So on the left, Francesca Squillante. On the right, Julie Waters. That's our school. That's the precinct. That's our connectivity. That's my picture to kind of lead up to genetically determined traits and how we study them. I, for many years, studied pretty much all my life neurodevelopmental disabilities or neurodisabilities, and I started with learning disabilities. And these affect 1 in 6 children or 1 in 20 adults of a lifetime. And not all of them are genetic, naturally. So we worked with intellectual disabilities, we worked with epilepsies, autisms, movement disorders, and most recently cerebral palsies, which were not considered to be genetic. For many years and in many jurisdictions, they are still more considered to be brain injury rather than a gene-driven group of disorders. You would be also quite impressed by the changes over the last 30 years in the number of genes which explain these disorders, as well as perhaps the impact on our societies. And these are just numbers from Australia as a consequence of these disabilities. So this diagram is complex on the left. It's a Venn diagram which shows you overlap of the gene, but it's an interesting structure. And you see on top in your forefront, like SCN2A, which is a known epilepsy gene, but it is also a known autism gene, it is a known intellectual disability gene, and it is a known movement disorder and cerebral palsy gene. So through this, I would like just to illustrate that even though the genes are identified as epilepsy genes, really the background impact errors in these genes made on individuals is a lot more complex than just the way how they were identified originally or how they describe in their name. And PCDH19 probably sits somewhere there. It's also one of the genes which was identified as epilepsy intellectual disability gene, but it's as much an autism gene or even cerebral palsy gene as we keep learning. So it is the elephant, if I can use the allegory, which is touched by 6 blind men, and they're trying to define what is it? Is it cerebral palsy? Is it epilepsy, or it's something behind that? I'm coming to this really from the genetic perspective, where we appreciate the genetic or the clinical diagnosis, but also look at the underlying genetic and cell and molecular biology drivers to better understand it, so we can better intervene and more meaningfully. So this is my history on the X chromosome over many years, and there are major epilepsy genes on the human X chromosome, and Steve Wilton nicely explained that men had 1 X and 1 Y chromosome, and women have 2 X chromosomes, and that's very important also in the story I'm going to tell you today. So we identified the ARX gene. We work with CDKL5 since the very beginning when we identified it. I'm going to tell you about PCDH19, but we also work with IQ-SEC2, for example, and I noticed there are parents or IQ-SEC2 or CDKL5 for that matter, or USP9X. So that's where we make mouse models. We work with patient groups, and for PCDH19, I was very fortunate really to walk the way and be part of the story from the initial identification in epilepsy as all the way through the models to providing proof of principle clinical trial. Now, out of interest, just on this 1 chromosome, there are probably about 100 genes which have associated seizures with them. So it's not a small number. There's only 1 chromosome. Another big picture view is that when I entered the field in '86, there were only 40 disease genes known. Now we have more than 4,424 for about 5,820 different diseases. Now, the first epilepsy gene is here. It was identified when I came here in '94, soon after, at the Women's and Children's Hospital here in Adelaide, in collaboration with Ingrid Scheffer and Sam Berkovic. The PCDH19 gene we identified in 2008, again, in collaboration with this famous Melbourne team. But you can also see that since 2008, there were about 3,000 different disease genes identified, and hundreds of them leading to different types of seizures. So I would like to play you a very quick video, which is a courtesy of Julie Waters and the PCDH19 Alliance, to introduce you to PCDH19 clustering epilepsy.
When Violet McManus was born, her parents were thrilled. She was their first child, a perfect little girl who met all of her developmental milestones.
She was learning to pull up, and she was saying mama and dada. It was just really, really great.
She was just the best thing in our lives.
Krista Pandaleos felt the same way when her daughter Olivia was born.
I was very excited I was having a girl. When we found out, we were beyond excited. Everything was fine with the pregnancy.
These children look very different, and their families may not have much in common. But before their first birthdays, one significant, life-changing similarity would emerge.
When she was 10 and a half months, she had her first seizure. We flipped on the light and she was completely blue, and there was bubbles coming out of her mouth. So then we called 911, and she had hundreds of seizures within 2 weeks of being hospitalized. She was never the same child after that. She stopped speaking. She didn't know who we were. She was gone. I couldn't really formulate what was happening. I'd never seen a seizure before in my life. And I just was panicking. So I called 911 and just said, please come quick. I don't know if she's going to die.
Over the years, doctors would try dozens of medications to control the girl's seizures, and their desperate parents agonized over what might have caused their child's illness.
Thank you for that. Thank you to Trista and Julie. So from this, what you heard, this is a type of epilepsy with normal or almost normal development until about the age of 8 to 12 months, when the first seizure clusters happen, usually associated with fever. And from there, there is all sorts of complications, clusters of seizures, and in some individuals, regression. So we're celebrating almost 50 years when this type of disorders was first noticed in the United States. And this is a large pedigree where you can see the circles are women, the squares are males, so you'll be all very familiar with it. And it captured the attention because it was special. What was exceptional about it, that only the women or girls were affected, while males were not affected. So this intrigued a generation of scientists and clinicians. And in 1997, using the same family, they managed, usually genetic tools, so-called genetic mapping, to place the likely cause of the human X chromosome, which was also quite exceptional because, as you heard from Steve, because males have only one X chromosome, usually they are the ones who carry the burden of the disease when something goes wrong on the X chromosome, because women have the second X chromosome, and they usually or generally is the substitute helper for the damaged or non-properly functioning other gene on the other X chromosome. What is also intriguing here that you see in these generations 3, there are 11 male offspring, 11 brothers, and almost every single one of them have affected daughters. Now, we didn't know where to look, so we pretty much looked through everything on the human X chromosome, and that's how we identify this molecule, which is a protocadherin 19, is a cell adhesion molecule, as we call it, which is somewhat responsible for our cells sticking together in our tissues, in our organs, but also for our nervous system and for our neurons to wire the brain together, so form the fibers of neurons, those bundles together, and that's how they communicate. So we identify this, and again, there was nothing special. So it's quite an exceptional that why this particular gene would behave so strangely in a way that men who don't have a properly functional gene or its product are not affected, and in opposite, women who have a spare copy and one copy which is not functioning properly are the ones who actually bear the consequences. Now, to some extent, the explanation came a year later from Christelle Depienne and her colleagues, where they identify this male with so-called post-zygotic somatic mosaicism. And they identify this patient in a group of SCN1A negative Dravet cases. And for a few years, the PCDH19 epilepsy was also referred to as Dravet-like. Also, now we know they are a very different group of epilepsies of early onset. So the post-zygotic mosaicism means that there is a sperm and an egg, they fertilize, and after that fertilization, at some point, one of the cells acquire an error in one of the X chromosomes. As such, these males become mosaics. So that's how we realize that perhaps this disorder only occurs when we have a mix of cells in a single individual, whether it's a male or female, with cells which function properly with PCDH19 and cells which do not have PCDH19, perhaps at all. It's an interesting molecule also from another perspective, but that's not for today's presentation, which somehow determines the differences in the brain between men and women. There are differences in size and volume and other features, and PCDH19 is one of them, who has been now identified as a responsible molecule. So this epilepsy is a disorder of mosaics. Just to explain it in a pictorial way, we have individuals like here on the left with a green central nervous system, which are the ones who have properly functioning product of PCDH19. And then we have those with purple central nervous system, and those are individuals who don't have perhaps this product at all, yet they are functioning properly, which is kind of intriguing, because we keep hearing from many other disorders that if something goes wrong with the gene and the protein, then we have a disorder. Not in this case, as it looks. So the things go not according to the plan when we have this kind of a mosaic central nervous system. And that can happen in women because one X chromosome is usually shut down in a particular cell, and as such, they end up being mosaics. So mosaics of cells which express the chromosome X from mom or the chromosome X from dad. And that creates this mosaicism, and that's why the central nervous system has this pattern, which is actually a picture taken from my colleagues' studies in a mouse model. So it's not made up. It's an actual picture, but it's a mouse brain put into the central nervous system image of a man or a woman. And that, of course, happens in the constitution of the post-zygotic somatic mutation male. Again, they have cells with normal X chromosome functioning, and they have the cells where the X chromosome is not good. So in a nutshell, it is, we have different types of good. It's a good with the gene, and it's a good without the gene. So what can go wrong? The wrong goes and happens when these two different type of goods happen in one individual, and that becomes not good. And that really made us scratch our heads quite a lot. How to even understand this, how this has happened, and not even how we can fix it. So we have many questions. Is this just really epilepsy, or there is more to it? How and if the different DNA variants get different outcome, because that's, for example, the case in SCN1A or Dravet spectrum or SCN2A as well, and many other questions. So with a student, Christy Colts, we have done a review of the literature, and what we found pretty much confirmed what you heard from Julie and Trista in the video, that really there is a period of up to normal development to the age of about 10 months to 12 months when the first really lightning strikes and the first clusters of seizures appear. But now we have numbers, so we have with that a probability And so we can feed back to the other patients and provide advice. In about 81%, these onset of seizures is associated with fever, and that could be also associated with immunization. And that's where sometimes there is an opportunity for misunderstanding that it was the immunization which triggered or caused the seizures. What we also have done, we looked through about 54 different girls and plotted the onset here in green and offset of their seizures against the hormonal levels, which are kind of the average in a population of male and female hormones. But what was really intriguing there, you see that after this mini puberty and about 5 to 6 months, we see this huge green peak of cumulative onset. That's when the girls start having seizures. So it somehow looks like when they become unprotected because of the mini puberty kind of weaning out, that's where the brain stresses due to stressors like seizures or others, or naturally the clusters of seizures happen. And the offset also intriguingly coincides with the onset of puberty. So in many cases, these girls, for those at least who know they are old enough, they stop having seizures when they go into the puberty. So that's also gave us an idea, maybe the hormones play an important role. And we know we do in different seizure disorders, catamenial epilepsy. So it was nothing really novel, but it was quite intriguing for this type of seizure disorder. And then Rebecca looked almost at 400 different cases and 22 males, because of course they are around, but they are less frequent, but they are not insignificantly frequent. They are perhaps not well recognized as such. And again, you see after the mini puberty, this huge peak, cumulative peak of onset when they start having seizures. But you also see that it's probably spread across the 20, 30 months of the first of their life. What was also important, what we found as part of this study, was that it perhaps depends whether you have a seizure before 1 year of age or after the first one, I mean the onset of seizures, and whether that predicts your outcome in terms of learning and memory tasks. And it does look like it's quite significant. So if you have seizures before 1 year of age, the outcome is usually more severe statistically, but not individually. And that is the devil in the detail. And if you have seizures after 12 months, usually the outcome is less severe. And you see it on the circus plot, where on the left you see the onset, and on the right-hand side of the dial, you see the outcomes. And you see the black, which is the onset after 12 months, goes predominantly to normal, borderline, and very rarely to severe, to profound outcome. And we found the same in a survey, which we looked at another 95 cases. So it's quite robust data replicated across a large number of individuals. Then we asked the question, so does it matter whether you have this particular DNA change or that change or this change? So we collected 20 individuals with this very specific change. So they are unrelated, but they all have the same driver of their PCDH19 clustering epilepsy. And you can see when plotted against this diagram, they can have onset before 12 months or after 12 months. They can be pretty much unaffected. That's up to 20% individuals just not affected, are not affected, even though they have the variant, or can be profoundly affected. And then we took another such a variant, which we find frequently, up to 30 independent times, and we saw the same picture. So in this case, we could tell, at least be confident, which is unlike like in SCN1A or SCN2A, where probably the variant type doesn't really matter in view of the outcome. But on the other side, even for these individual variants, we see very variable outcomes. So again, scratching our heads, so how are we going to resolve this? And maybe I will have a solution for you towards the end of the talk. So the question then was, is this onset of seizures also causing other problems, or can we correlate? Now, initially, this was described as seizure disorder with learning disability. Nobody was talking about autism. Now we know at least 60% plus individuals have autism spectrum spectrum disorder, ADHD, and OCD. And here, Christy put the similar data together, suggesting that if you get the seizures before 12 months, the outcome in terms of ASD measures is more severe than if you get the seizures after 12 months. Again, it's a statistical measure as a group, but not necessarily applied to specific individuals. So we look at different measures because we really want to understand this when we want to intervene. This is not just a seizure disorder. This is a very complex neurocognitive and neuropsychiatric disability, which we need to pay attention, and probably the outcomes in terms of these measures will link to different structures of the brain, maybe different developmental stages where we should think and consider in terms of intervention. So in putting it in a pictorial way, we kind of want to know when we have a PCDH19 variant, which way the individual likely will go. Are they just going to have a few seizures and then they will be fine? Are they going to have full-blown severe clustering epilepsy? And that's we really don't have anything to really predict it at the moment. So we also found with Christy that this is just definitely not just a seizure disorder. There are all sorts of other issues, and you see it quite frequently, 50 to 70 to 85% of prosocial deficits, conduct problem, emotional problems, aggression. So these are type of studies, of course, which are very useful for us to understand the underlying basis of the outcome in patients. What we also did was very interesting, because you may ask, I mean, because it's so complex in the genetics, are these affected males and affected girls kind of similarly affected, or there is some difference? And it looks like they are very similarly affected when you look at the blue and the purple line here. That's a combined score across different measures of the neurocognitive and neuropsychiatric features. They kind of track together. Now, the other question was that are really those individuals who have this PCDH19 variation and are not affected, because the penetrance is only about 80%, are truly unaffected? And again, using these measures, we can see that the red line tracks nicely with the green line, and the green line are the unaffected males who are, if I use the word, knockouts, or are just deficient for this gene product. I just don't have it, and yet they are unaffected. And just to show you this pictorially, that it's not like one discriminatory value above and below, but it's a spread. And this green box here on the left-hand side, it's the spectrum where we consider it unaffected space. And you see there are a few individuals who fit there. And then there is the spread of different measures all over. So it's a continuum, really, and almost every single patient has a very specific phenotype, which again, it's going to be quite challenging if we're going to try to correct it. So this is what we face. We know, and perhaps in future we will be testing these individuals at birth, and we'll identify loss of function of this important molecule. So what we're going to be able to tell the parents and the health services, what the outcome is going to be. So we know that these are the percentages of the outcomes in terms of cognitive, autism, other behaviors, or non-penetrant, about 20%, but we don't really know what is really pushing the outcome of these individuals to different category. So we think it is the level of the mosaicism. So I told you that you have mostly central nervous system where the majority doesn't have functional PCDH19, you almost end up without a problem. If you have a wild type situation and very few cells would have missing PCDH19, that's also compatible with very mild outcome. The biggest problem happens when it's almost 50-50%, when it's the perfect mosaic. Now, with colleague Paul Thomas, we have nicely confirmed this is a mouse model. So these are mouse identical twins and they brains. So for all purposes, these are identical twins, genetically identical. They have deficient PCDH19, and those are the cells which are in purple, and properly functioning PCDH19, which is the color in green. And you can see that all these female twins in mice have different pattern of the purple and green. And that we found related to their seizure activity. So to that matter, we also have at least 6 monozygotic, that is genetically identical twins, where both have PCDH19 variants, and generally one is less affected or unaffected versus the other one. And that's where we also study these to understand that what is driving really in one twin to have the disorder and the other one not, or one twin have a much milder form, because simply learning from that, again, we perhaps can put strategies in place to understand how to manage it better. And again, interestingly, the electrical activity here in the middle, only in the mosaic type, is much more extensive, and that's courtesy of Steve Petra and his team at the Florey in Melbourne. Now we reproduce this on a dish. So here you have a good cells, then you have the cells with the variant PCDH19, and then you have the cells which are mixed, and you see that almost look different. They form their clusters differently, just under the microscope. But when you look at the activity, they really have seizure on a dish, which is good to know. It's a great model where we perhaps can look at screening of different types of drugs to perhaps subdue a little bit of this activity and see how it can translate through the mouse models to human situation. So how we fix it? There is very little known about treatment of PCDH19 clustering epilepsy. There are published studies with success here and there with corticosteroid therapy, stiripentol, or clobazam or bromide, probably from the biggest review of the literature in terms of anti-epileptic medication. Lynette and Ingrid, together with Christy, has led perhaps the most recent study of levetiracetam or Keppra in PCDH19 with an interesting data in a group of 17 female. The success was pretty good. 76% became seizure-free after 12 months and 62% for more than 24 months out of these numbers. And in the cohort B, which Christy collected through the survey of the international PCDH19 community, we saw the effects kind of tracking similarly, but perhaps smaller percentages. And the published study also found that 5 out of 38 cases were seizure-free on Keppra as well. So these are just some of the examples of the seizure diaries and the introduction of Keppra in the study of Lynette, Ingrid, and Christy. And here, just to remind you that this is unfortunately also the time after about between 5 or 6 years and about 20 years when we see the onset and offset seizures happening. So it is really the question there, well, are these seizures stopped because of the drug or they stop naturally? And for that, of course, we need large cohorts and more information. So we also took a more data-driven approach, and we found that these girls were deficient in particular hormone. And in this case, a hormone which is active in our central nervous system, and that's why we call them neurosteroids. And here in particular, we looked at allopregnanolone. So in brown or dark red, you can see the effects on the affected girls, and in blue are controls, and they are significantly different. And then we linked this molecule to estrogen receptor, and that was again another exception. How a cell adhesion molecule will interact with the estrogen receptor, which is known as a regulator of gene expression as a consequence of steroid hormone. But this finding was replicated by our colleagues in Italy we worked with, and they, again, nicely on a cohort of 24 individuals demonstrated that these girls are indeed hormone deficient, and specifically allopregnanolone, and even more pregnenolone sulfate, which is one of the modified neurosteroids acting in our brain. So the first reaction was based on, since 2014-15, to really think about a hormone replacement therapy technically. And there was fortunately available ganaxolone, and many of you probably would have heard about ganaxolone. It was trialed by Marinus. That's the only company holds the intellectual property to ganaxolone, which is a modified version of allopregnanolone, acting on the GABA receptors and doesn't have the hormonal effect. So it's modified. It's not the same as allopregnanolone. And there was some excitement about this, perhaps bringing help to this group of disorders. So the latest really preliminary data of the Violet study, which Marinus released, showed that ganaxolone demonstrated a median 61.5% reduction in seizures compared to about 24% reduction in the placebo group. But that data is still being analyzed, and we are part of it. And they also looked at other measures of the outcome, not just the seizure frequency, but also the neurocognitive and neuropsychiatric outcomes, to really take it more in entirety rather than take it by one particular measure to see the impact of the treatment using this hormone replacement strategy. And you may know that perhaps in CDKL5 epilepsy, where we don't know how CDKL5 relates to PCDH19 in the cell level or at the molecule level, interestingly, ganaxolone seems to be quite effective as part of the Marigold study in CDKL5. Perhaps even the data is perhaps even more exciting than for PCDH19. So in principle, the treatment for this particular disorder, we can do the hormone replacement, and that's based on the opportunistic findings, data-driven, when we found that these individuals are hormone deficient. But neurosteroids are really being speculated to be used for treatment of epilepsies for many years, and now they're also becoming a target and opportunity for intervention in autism spectrum disorders. And the top 8 autism genes have been linked to estrogen receptor signaling in the latest studies, which is showing that there is perhaps a common pathway we might be able to target to bring some improvements in different compartments of the neurocognition or learning of epilepsies in this group of disorders. But for PCDH19, of course, given that it's a disorder of mosaics, we can look at reintroducing the PCDH19 for those who don't have it, or perhaps ablate even the normal copy in those individuals who are mosaic. And those studies are ongoing, and we don't know the results, but technologies like Steve Wilton presented or Steve Petru presented last year are highly relevant to this group of disorders. The question is when to treat. So this is a disorder of a lifetime, or of a lifespan, I should specify, where we don't see major problems early in development. We see the onset seizures during the first year of life or soon after. They usually stop in puberty. So this is the time where really we see the major seizure activity, but the neurocognitive decline pretty much doesn't get better with puberty. And then Ingrid and her colleagues also showed that perhaps there is more complications for later life for older individuals with seizures reoccurring and more complex psychiatric problems coming up as well. So perhaps we really need to intervene during the first year of life, and ideally before the first onset of seizures. For that, we need to know which individual is going to have the seizures and which not. So we work on it, and this is my last data slide, where I think it's quite exciting that we can stratify these individuals, those who are affected, who are not, based on these markers, And without knowing their seizure history, we can tell whether they are affected or not. Now, the question was whether we can tell those who have the variant and are not affected. And it looks like we can. So I'm hopeful here that in those individuals who perhaps in future will be diagnosed with this type of epilepsy, even before they first seizure onset, we'll be able to tell whether they have the seizure disorder or they are likely to be non-penetrant. Because if we can avoid aggressive treatment, that's also a huge benefit to these individuals. But that's really a research which still needs to happen and needs to be refined before it's clinically applicable. So it is an exceptional disability which showed us that certain genes are maybe disposable for individuals, for males, and perhaps also for females. Also, we don't have females born without 2 copies of PCDH19. This is an interesting disorder, cellular mosaic. Whether it's unique, we have to wait and see, but it has taught us about different goods which can be in our cells, and that's how we assess what is good and what is not so good, and which of course applies to other types of epilepsies. And of course, as you heard, this is not just an epilepsy. This is quite a complex neurocognitive, neuropsychiatric problem. And that's probably about it. I just would like to thank all my colleagues, national and international, and I would like to say thank you to this community. I mean, without their contribution, motivation, with these lovely girls I get to see pre-COVID probably once a year, whether in US or in Europe, I probably wouldn't be doing what I'm doing. So thank you, everyone. Thank you for listening, and thank you for pushing us scientists forward to find a solution for you.
Jozef, thank you so much for your fascinating presentation. And I find myself having a couple of questions, and there are also a few from our audience. One question that I have heard this question many times in our community. We know that these genetic epilepsies are usually, the word epilepsy is only a piece of the disorder. And that's something that you pointed out as well. Often, not always, often the seizures are the worst, but often for families, it's the other problems that pose the most comprehensive difficulties. And yet, when testing and trials come through, often they're centered on the seizures because it's measurable. And you can imagine the challenge that this poses for families, because that's may or may not be what they're really interested in. Your work makes me wonder if you see the potential for there to be other markers that we can use as we try to determine what kinds of medicines, what kinds of therapies and protocols might help our kids.
Exactly right, Sarah. Just try to be concise. Yes, the PCDH19 community is very much concerned about the other outcomes and not just seizures. Seizures, they know they come and go. So that's why we designed the survey. That's why we looked into how we can measure the other outcomes, and those measures were implemented in the clinical trial. And anecdotally, from the previous trial, actually, these patients done really well on ganaxolone in terms of the non-seizure outcomes. Their behavior improved, their attention improved, but again, it's on a case-by-case basis, so we don't have yet robust data, but we do look into it consciously. And even from the mouse model perspective, it looks like that if the seizures are perhaps triggered from the cortex, maybe the learning and memory outcomes are triggered more from other structures of the brain, like the hippocampus. And for example, the zebra pattern you saw in the cortex, it's actually not happening in the hippocampus. So it looks like that even though we see different outcomes, maybe these are also driven from different structure, different types of cells within the central nervous system. So we're aiming at understanding it, we're aiming it and pulling it out and really targeting it. So it is going to be, and like in cancer, it's going to be a multi-pronged approach where we're going to be using perhaps a combination of drugs, maybe at different times, to manage these complex and neurodisability disorders, and not just seizures.
I apologize. I didn't mean to interrupt you, but I was going to say I'm actually glad you mentioned cancer, because I'm old enough to remember going back to a time when, you know, cancer treatments and new cancer therapies, my goodness, they were happening exponentially, and they were quite striking and in many cases surprising. That's still happening, of course, but there was a lot of money, a lot of energy, you know, a big aura all around that, a bit of a halo effect as well. Do you think that can happen for genetic epilepsies and this community? And what does it take to make that happen?
Sarah, you tickle my nerves. I usually say these days we should consider these neurodevelopmental disabilities as urgent as, for example, childhood cancers. We can't say that, oh, let's wait what happens and whether it gets better. We know that we need to intervene early as possible and meaningfully. We need to dare a little bit more. We saw how dramatic advances being made in blood cancers in particular, especially in childhood onset, especially informed by genetic findings. So I think the urgency is there to consider and convince the funders and everyone, and this is a fantastic community to help us to convince the policymakers and those who distribute these funds to distribute them more equally, because cancer is important, we relate to it, but now we also relate to these disabilities which deserve equal attention. I can't say more, 100% agree.
Well, I'm glad to hear that, and we appreciate your support on this issue. And now here's one that we have coming in for our community. Some of the DEEs, the developmental epileptic encephalopathies, have been divided into gain and loss of function variants, and you touched on this. But within those larger groups, there are a lot of variations. So how could your techniques and what you're studying there be used to gain more insights into these differences in clinical presentations, and again, eventually with an eye toward therapies and treatments for people who are suffering from these various epilepsies?
So this is an important question. Of course, it is kind of a holy grail to find a treatment for all outcomes, irrespective of the time we intervene at. My take on it is that there will be a type of epilepsies where fixing the gene which is broken will fix the problem altogether. And there are types of epilepsies, or what we see as epilepsies, where there is problem which started in utero and it's more complex. And fixing seizures will require drug A, fixing memory will require drug B, and fixing behaviors, drug C. So from this perspective, the pathway we study, that provides kind of the endpoint help, which will be probably applicable across many group of disorders, because we know autism is really tapping into the same pathway. So again, we know, for example, estrogen in ARX mouse model, we can ablate the seizures, but we do not ablate the learning and memory problems. So that's telling us that we do need to persevere with investigating these pathways, because it's going to be a helper at one front, but maybe certain epilepsies will require additional drug in combination to manage the outcome. So we need to have both very, very variant and gene-specific interventions, but we also need to have those interventions where all these different types of epilepsies collide and perhaps cause dysregulation of oxytocin receptor, all these neurosteroids in the blood, and as such, we assume also in the brain. Also, that's not easy to measure at the moment. So it is a complex strategy which we call precision medicine, but we are kind of hiding behind the unknown as yet, or not that well known.
Well, it's a fascinating area, and we are delighted to have you participate this year and to hear your insights about genetic epilepsies, and these ones in particular. But thank you again, Jozef Gecz, for joining us from South Australia.
Thank you.